Shadows Beneath the Crown, Uncovering the True Origins of the Black Death. Introduction. Not the rats, not the fleas, then what? It began with whispers, ships arriving at Sicilian ports carrying men with blackened boils, coughing blood, and dying within days. It was 1347, and within 5 years, nearly half of Europe's population would be gone. Villages turned to graveyards. Entire families vanished overnight. The dead were carted through city streets like refuse, and the living huddled in terror, convinced the end of the world had arrived. This apocalyptic event would later be known as the Black Death, and it remains one of the most catastrophic pandemics in human history. For centuries, the dominant explanation pointed to rats and fleas as the main culprits. Yersinia pestis, the bacterium blamed for the plague, was said to be transmitted from rats to humans via flea bites. The story was tidy, satisfying, and scientifically backed until cracks began to appear. Recent research has challenged this narrative, suggesting that the truth is far more complex and possibly far more terrifying. 1. The traditional narrative, a tale of rats, fleas, and bacteria. For centuries, the cause of the Black Death was thought to be straightforward, the bacterium Yersinia pestis, carried by fleas that lived on black rats. This narrative has been entrenched in scientific literature, documentaries, and historical accounts to the point where it became accepted as the single truth about the plague. The theory emerged in the 19th century when bacteriology began to evolve and scientists, like Alexandre Yersin, isolated the bacterium responsible for bubonic plague. The plague's gruesome symptoms, sudden fever, swelling of the lymph nodes, buboes, and the painful darkening of the skin, seemed to line up perfectly with the signs of Yersinia pestis infection. Fleas, living on rats, were believed to bite the rats, ingesting the bacteria and then passing it on to humans. According to the classic model, when a rat population was wiped out by the disease, the fleas, desperate for a blood meal, would turn to humans, causing the disease to spread rapidly. Rats were the focal point of this model. Those rodents were seen as the primary vectors, carrying the deadly disease across Europe via trade routes, particularly through the Silk Road, which connected Europe and Asia. As ships carrying goods docked at various ports, the rats would disembark, spreading their fleas and the bacteria far and wide. This model became widely accepted and was further cemented by the fact that plague outbreaks in history often coincided with the movement of rats and fleas from infested ships or trade routes. The familiar image of the black rat, Rattus rattus, fleeing from the ports, and the oriental rat flea, Xenopsilla chiopus, transferring the disease to humans, seemed like a logical and concrete explanation. Additionally, Yersinia pestis was indeed found in the remains of plague victims across Europe in the 14th century, adding scientific weight to the idea that the bacterium had a critical role in the spread of the disease. It could be cultured in labs, and its ability to cause plague-like symptoms in lab animals reinforced the model. By the 20th century, this theory became gospel, shaping not only how we understood the Black Death but also how we conceptualized pandemics. 2. The timeline that doesn't add up. For years, historians and epidemiologists accepted the rat-flea bacteria model of the Black Death without much question. But as researchers began revisiting primary sources, digitizing historical data, and applying modern epidemiological modeling to 14th century events, inconsistencies started to emerge, particularly regarding the speed and geographical spread of the disease. The bubonic plague, as caused by Yersinia pestis, typically has an incubation period of several days, and its primary transmission vector, fleas, require time to infect a new host. In modern outbreaks, such as those observed in India or Madagascar, the spread of bubonic plague is slow and localized, often contained within households or small communities. However, the Black Death in medieval Europe behaved nothing like that. According to historical chronicles, the disease could travel up to 30 kilometers per day, decimating entire cities in a matter of weeks. One of the most cited examples is the rapid progression of the plague from Sicily to Paris within the span of months, without any known rat infestations large enough to explain it. In England, it spread from the port of Melcombe Regis to London and the countryside at an astonishing pace. This epidemiological contradiction has led scholars to ask a crucial question: could fleas and rats alone really have carried the plague across continents so rapidly and efficiently? Moreover, many medieval reports describe entire households and even entire streets being infected within a day or two of the first case. That pattern more closely resembles diseases that are spread by respiratory droplets, such as influenza or pneumonic plague, than by flea bites, which typically result in more staggered and limited infections. Another problem lies in seasonality. Flea activity is heavily influenced by temperature and humidity. In theory, plague outbreaks should diminish during the cold European winters when flea populations dwindle. And yet, historical accounts show waves of Black Death occurring even in winter, defying expectations for flea-based spread. There are also few consistent descriptions of widespread rat die-offs, which should have preceded human outbreaks if the rodent vector theory held true. Mathematical modeling has further destabilized the traditional narrative. Simulations based on rat flea transmission rates cannot account for the explosive outbreak patterns seen during the pandemic. Instead, models based on human-to-human transmission, possibly through airborne droplets, match historical data more accurately. What's more, the lack of immunity among affected populations is striking. If the plague had been spreading slowly via rats and fleas for centuries prior to the 1340s, as some theories suggested, then why were populations completely unprepared immunologically when it struck? As these inconsistencies mounted, it became clear either the pathogen behaved very differently in the past than it does now, or we were looking at the wrong mechanism entirely. 3. Genetic clues buried in the soil. When the 14th century world was consumed by the Black Death, there were no microscopes, no laboratories, and certainly no gene sequencers. But centuries later, as modern science began exhuming the past, sometimes literally, the buried dead began to speak. In recent decades, a powerful new tool has emerged: ancient DNA, DNA analysis. Scientists began extracting genetic material from the teeth and bones of plague victims buried in mass graves across Europe. These samples provided the first concrete proof that Yersinia pestis was indeed present in many victims of the Black Death. One of the most famous discoveries came from the East Smithfield Cemetery in London, a burial ground created exclusively for plague victims. In 2011, researchers successfully extracted Y. pestis DNA from several individuals, confirming the bacterium's presence in 14th century Europe. Similar findings soon emerged from plague pits in Germany, France, Sweden, and Italy. On the surface, this seemed to validate the traditional model, but the details told a different story. The strain of Y. pestis found in these medieval remains differs genetically from modern strains. It is now believed that the Black Death was caused by an extinct or nearly extinct branch of the bacterium, one that may have had unusual virulence or transmissibility. Some variants show gene deletions or mutations that could potentially enhance their spread through respiratory means, although this remains controversial. Even more intriguing were findings from Central Asia. In 2022, a study published in Nature traced the origin of the Black Death to the Tian Shan region near modern-day Kyrgyzstan. At a medieval burial site near Lake Isyk-Kul, gravestones dated 1338 mentioned a mysterious pestilence. Scientists analyzed teeth from these graves and found Y. pestis DNA that appears to be the genetic ancestor of all later Black Death strains. This Central Asian origin aligns with historical speculation about trade routes like the Silk Road being the highway of death. But it also raises more questions. How did a localized outbreak in a remote village explode into a pandemic that leveled Europe in a few short years? Another twist came with the realization that multiple strains of Y. pestis may have been circulating simultaneously. Genetic variation among victims in different European sites suggests that the Black Death may not have been one monolithic event, but a series of overlapping outbreaks, possibly driven by different mutations of the same pathogen, or even by multiple pathogens acting in concert. Some scientists now believe that the virulence of the medieval Y. pestis strain was so severe that it could potentially support airborne transmission, especially in the form of pneumonic plague, which infects the lungs and spreads via droplets. This could explain the observed speed and mortality rate that modern bubonic models fail to replicate. Despite these advances, genetic evidence still can't answer every question. DNA is notoriously fragile, and the picture it paints is often incomplete. Yet with each tooth unearthed and each genome sequenced, we edge closer to understanding how the Black Death may have truly operated, not through simplistic contagion, but through a biological complexity that even 21st century science is still unraveling. The graves of the past are not silent. They whisper secrets we are only just beginning to hear. 4. Was it pneumonic plague all along? What if the terrifying swiftness of the Black Death wasn't due to flea bites or contaminated rodents at all, but something far more immediate and insidious: airborne human-to-human transmission? For decades, this question lingered at the fringes of plague scholarship. But mounting historical, clinical, and genetic evidence is pushing it into the mainstream. Increasingly, experts are asking, was the primary killer not bubonic, but pneumonic plague? Pneumonic plague is caused by the same bacterium, Yersinia pestis, but manifests differently. Unlike bubonic plague, which involves the lymphatic system and is usually transmitted through flea bites, pneumonic plague infects the lungs. It spreads not through vermin, but via droplets expelled by coughing or sneezing, similar to influenza or COVID-19. And it kills with shocking efficiency. In untreated cases, mortality approaches 100% within 24 to 72 hours. When medieval chroniclers wrote of the Black Death, they described a disease that moved like wildfire. People fell ill in the morning and were dead by night. Entire households were wiped out before a physician could arrive. In some cities, death followed exposure within a day or two, a timeline that doesn't fit with the flea-based bubonic model, which requires an incubation period of 2 to 6 days. One of the most famous accounts comes from the Italian writer Giovanni Boccaccio, who in the Decameron wrote, "It showed its first signs in men and women alike by means of swellings in the groin or under the armpits. But later these grew in size and appeared on any part of the body, and soon after, the victim would begin to cough blood and die swiftly." While Boccaccio mentions buboes, his description of coughing blood, a hallmark of pneumonic plague, is telling. Other witnesses describe victims collapsing in the streets, their bodies darkened by internal bleeding, with little to no visible swelling. Priests performing last rites often died within hours of visiting the infected, suggesting close contact transmission. Further supporting the airborne theory are plague outbreaks in colder months, when flea activity would be minimal, but when people remained indoors, breathing shared air in confined spaces. In modern outbreaks of pneumonic plague, such as the 2017 epidemic in Madagascar, authorities saw the disease spread rapidly in densely populated urban areas, often transmitted through casual conversation or shared public transport. Moreover, recent genetic studies suggest that the medieval strain of Y. pestis may have possessed mutations that enhanced its virulence and transmissibility. Some variants appear to have an elevated capacity to survive in lung tissue and promote person-to-person transmission. Though this research is ongoing and highly technical, the implication is stark. The Black Death may not have been just bubonic, it may have been pneumonic or a hybrid of both. Another theory proposes that a dual outbreak occurred, bubonic plague carried by fleas, and pneumonic plague spread via the air. This could explain the varied symptoms and transmission speeds described across regions. In some places, where rat populations were dense and sanitation poor, bubonic plague may have prevailed. In others, particularly colder, more urbanized environments, the disease may have gone airborne. But why does this distinction matter today? Because how we interpret historical pandemics shapes how we prepare for future ones. If the Black Death was capable of sustained airborne human-to-human transmission, then it serves not only as a lesson in mortality, but as a chilling prototype for modern pandemics. It reminds us that pathogens can evolve quickly, behave unpredictably, and leap from ecological reservoirs to the lungs of cities with devastating origin. Long before the Black Death engulfed Europe, a quiet storm was brewing thousands of miles to the east. In the mountains and deserts of Central Asia, caravans laden with silk, spices, gems, and porcelain wound their way westward along the Silk Road, the legendary network of trade routes that connected China to the Middle East and beyond. But these caravans may have carried more than just wealth and wonder. They may have been the first harbingers of the deadliest pandemic in recorded history. Archaeological and historical evidence increasingly points to Central Asia, specifically the area surrounding Lake Issyk-Kul in modern-day Kyrgyzstan, as the likely cradle of the Black Death. Inscriptions on tombstones from the 1330s, written in Syriac script, reference a devastating pestilence that swept through local communities. These inscriptions, combined with genetic data from human remains in the region, have led many researchers to identify it as ground zero for the pandemic. The Silk Road, though celebrated for its cultural exchange and economic significance, was also a superhighway of disease. Merchants, soldiers, pilgrims, and explorers traveled vast distances, mingling with new populations and ecosystems. The combination of human movement, food storage, pack animals, and dense settlements created the perfect conditions for an emerging pathogen to thrive.